everyone. We are going to learn enzyme structure, function, kinetics, and inhibition today. So, what are the enzymes? Enzymes are the biological catalysts that accelerate the rate of biochemical reactions without being consumed during this process. So, a typical reaction, a substrate will be converted into a product and it remains the same, but an enzyme accelerates the reaction rate. So, it accelerates the process of this conversion of substrate into product, but it's not being consumed in this process. So at the end of the reaction, we'll have our enzyme back in its original form. So most of our enzymes are protein in nature, but we also have enzymes like ribozymes, which are RNA in nature. And this tells us the significance of RNA molecule because it's an only macromolecule which can act as both genetic material as well as an enzyme. So let's have a look how an enzyme accelerates the reaction rate. So we have on the x-axis the reaction progression of the reaction, and on the y-axis we have this energy. So if we draw a typical reaction, it would be something like this. The substrate will be somewhere here and the energy level, and in order to get converted into the product, it has to overcome this energy barrier, which is called the activation energy. So the transition state, which is an intermediate state during any reaction process, is a high energy state. So the reactants normally do not have enough energy to overcome this barrier, or only a very few substrate or reactor molecules have enough energy to overcome this activation energy barrier and reach transition state and then get converted into products. So that's why the reactions without an enzyme or a catalyst are very slow, so may take years and years to be completed. What an enzyme does, it lowers this transition state. So with an enzyme, with the presence of enzyme, the curve would be something like this. So the reactants are converted into products and they are attaining the transition state, but now this transition state is at lower energy level compared to the one which was without an enzyme or a catalyst. So this activation energy has been lowered, and this way we have more reactants available, which are present, which have this enough energy to attain this transition state and then get converted into products. So an enzyme actually stabilizes or lowers the transition state energy. It lowers the activation energy. But look here, the energy difference between the products and the reactants or the substrate remains the same. So there is the net energy released during this process, this react, remains constant. It's only the activation energy which differs between a reaction catalyzed by an enzyme or a reaction without an enzyme. So now, structure of an enzyme. So what kind of a structural component an enzyme would have? So one of the most important structural component is an active site. So active site in is a site which binds to the substrate or the reactant molecule, and it catalyzes the reaction. And then we also have this regulatory site. So at the regulatory site, either the activator or accelerator would bind, or an enzyme inhibitor would bind, and they would actually regulate or affect positively or negatively the enzyme activity. So each enzyme has an active site, but naturally enzymes also have the regulatory sites, because reactions in our body or in any living system are very controlled. So these regulatory sites helps bodies or systems to regulate the enzyme activity. So it will be either, if it's needed to more enzyme activity, it might accelerates the reaction through the binding of some regulatory molecules, or if the enzyme activity needs to be lowered or stopped, an inhibitor or a negative effector would bind on the regulatory site and inhibits the activity or lowers the enzyme activity. So these are the two key important sites or the structural component which an enzyme has. One is active site and the other one is regulatory site. So in 1894, Emil Fischer proposed a lock and key model. This lock and key model explains how the active site on an enzyme binds to the substrate. So this states that there is a structural compatibility or complementarity between the active site and the substrate structure in such a way that it acts as a lock and key model where the substrate actually has a structure which fits in to the complementary structure of the active site. And when the product is formed, the product would be released and we'll have our enzyme back free and the active site remains intact. This model is talking about a fixed conformation of the active site and a fixed concentration of the substrate which does not change over time, or at least it would does not change upon the substrate binding. So the active site has a certain conformation which is complementary to the structure of the substrate, and upon the binding of the substrate it doesn't change. It's just as a fixed structures and both fits into each other like lock and key. However, later on when the experiments were analyzed at different stages of reaction, for instance, when the enzyme was not bound to the substrate and when the enzyme was bound to the substrate, somewhat like this stage, and at the end of the enzymatic reaction, maybe somewhere here, they came to know that this stage looks like this in the real experiments. So it was something like this at the beginning, but upon substrate binding, it actually changed. And so this complementarity concept doesn't fit in, and the substrate is not something exact complementary to the active site in terms of the structure. So that means active site has changed its conformation. So without an enzyme bound to it, it was some substrate bound to it. I'm sorry. Without an substrate bound to it, the active site was something like this. But a sudden a substrate binds on the active site, it changed its conformation. This led to the proposal of the induced fit model, which states that the amino acid residues or the residues present at the active site, when they interact with the substrate or a reactant molecule, they actually change their conformation or change the conformation of the active site to actually fit in or to bind with the substrate molecule, which is not the exact complementarity in terms of structure to the active site in here. And this actually induces the interaction with the substrate, actually induces the conformational change in the active site, and then it adapts and binds with the substrate. And when a product is released, the enzyme is restored back to its unbound conformation. So the active site would be restored back to its unbound conformation, ready to bind to the new substrate molecule. So this kind of explains the native property of enzymes or the native property of any protein molecule, that it's not a fixed kind of conformation. Instead, it's kind of the upon the interaction, the conformation of the molecules changes or the structure changes, and it can actually adapt a different conformation. So to accommodate the substrate molecule, right? So this is how an enzyme interacts and binds to its substrate. Now let's have a look on the cofactor and coenzyme. So we know that enzyme catalyzes the reaction, but some of enzymes need additional components which are not activators or accelerators or inhibitors. They are actually the component enzyme needed for enzyme to catalyze a reaction. So they are called as cofactors and coenzymes. So an enzyme which needs a cofactor would be inactive without its cofactor components, and it's called as apoenzyme. When an inorganic molecule, which is called as the cofactor here, binds to the enzyme forms the active complex, and it's called holoenzyme. Now this enzyme is active. The holoenzyme is an active form of an enzyme, and most of the inorganic cofactors are metals in nature. For example, the DNA polymerase needs magnesium, and carbonic anhydrase needs zinc ions as a cofactors. So if you ever come across some PCR reaction, and if you've got to add magnesium, the reaction wouldn't proceed because the DNA polymerase need this cofactor magnesium for its activity, right? Okay. Some of the cofactors or the components which an enzyme needs, they're organic in nature. These cofactors are called as coenzymes, and same goes here. The enzymes which have organic cofactors or coenzymes, without the cofactor they would be in their inactive form, which is called as apoenzyme, and with the cofactor they form a complex which is called the holoenzyme, and holoenzyme is the active form of the enzyme. Whole enzymes with organic coenzyme have two different forms. Some of the whole enzymes which have organic component with them, the coenzymes, their organic component is called as prosthetic group because it remains tightly bound to the enzyme. So it remains bound to the enzyme, and even at the end of the reaction, the coenzyme would not leave the enzyme. So it would stays with the enzyme permanently. The examples are flavin nucleotides or the heme groups. So they are the component of the enzyme. So they are non-protein component. They are not protein in nature, these coenzymes. They remain covalently and permanently bound to the enzyme molecule, and that's why they're called prosthetic group. There are other organic coenzymes which are non-protein in nature, and they actually bind to the enzyme, but then at the end of the reaction they released, and the enzyme would be back in its apoenzyme form at the end of the reactions, and these coenzymes are called co-substrates. So, for example, coenzyme A, NAD, and FAD, they are the co-substrate for many enzymes in our body. So remember this thing. So the co-substrate are the coenzymes which are released after the reaction, and prosthetic groups are the coenzymes which are remain permanently bound to the enzyme molecule even at the end of the reaction, or when the enzyme is not catalyzing a reaction, they remain attached to the enzyme. And these coenzymes, these organic components, are non-protein in nature. So they are not protein molecules. The enzyme is a protein molecule, but these coenzymes, they are organic in nature, but they are not protein. So that's like both prosthetic group, co-substrates would be organic molecules, but they are not protein in nature. The enzyme is generally protein in nature, with the exception of ribozymes. Okay, so I hope now it's clear. Here is a summary of this coenzyme and co-substrate thing. So we have enzymes. Some of the enzymes are simple protein in nature, and they does not need any additional components. They are active without any kind of inorganic or organic component. So they are proteins, so they can function without any cofactor and coenzyme. Then we have other enzymes which need to form this complex or holoenzyme, in which one part is protein and other part is non-protein. So enzyme without its its in or non-protein part would be called as apoenzymes, and it's the inactive part, it's the protein part, and the non-protein part is called cofactor. If the cofactor is inorganic, it's normally called as cofactor, and it binds to the apoenzymes and actually catalyzes the reaction. The coenzyme are the cofactors which are organic in nature, and they themselves have two categories. One is prostatic group. The prostatic group involves the organic cofactors, but which remain permanently attached to the enzyme even when the reaction is be completed. On the other hand, we have coenzymes which are called as co-substrate. They are also organic in nature, but they actually bind loosely to the enzyme molecule or a protein component, and they are actually released at the end of the reaction. Enzyme kinetics. Enzyme kinetics is a study of the rate of enzyme catalyzed reactions. So let's start enzyme kinetics with the definition of KCAT. So in a normal reaction, our substrate molecules will be get converted into the product, and enzyme would be there to accelerate this reaction. So KCAT, which is also called as turnover number, can be defined as the number of substrate molecules converted to the product per enzyme molecule per second. So this is how you can actually define the turnover number or over number or KCAT. So this kind of tells us how efficiently and how enzyme is actually accelerating the rate of a reaction. Alright, so here we have a scheme which describes the enzyme catalyzed reactions. So in the first step, which is a reversible reaction, enzyme forms a complex with the substrate, which is called enzyme substrate complex, and then this enzyme substrate complex move to product, and the enzyme is then released to catalyze another reaction. And then K1, K minus 1, and K2 represents the rate constant for each reaction. So now if we on the y-axis we take the substrate, and on the x-axis we take the time, so over the time the concentration of the substrate would go down. Similarly, if on the y-axis we have products and on x-axis we have time, the substrate or product concentration will increase in a hyperbolic manner over the period of time, and the velocity of these reactions can be determined by the slope of each curve, which would be the rate of reaction. So change in the concentration of substrate over time or change in the concentration of product over time would give us the velocity of the reaction, right? So now if we take the velocity on y-axis and substrate on x-axis, so as the substrate concentration increases, you would say the velocity would increase, but then after a certain substrate concentration, it would remain constant. So the graph would plateau. So this velocity at which the substrate concentration would no more affect the rate of reaction is called Vmax. And using this graph, we can calculate the half of the Vmax. And this gives us a very important value which defines the efficiency of, which can be used to define the efficiency of an enzyme, is called Km. So the Km is a substrate concentration required for an enzyme to achieve half of its Vmax. It's also called Michaelis-Menten constant. And lower the Km value for an enzyme, the more efficient an enzyme would be. That means it needs low concentration of the substrate to achieve its half of the Vmax. Alright, so remember the Km. Km is not the velocity. Km is a substrate concentration which is needed to achieve half of the Vmax for any enzyme, and it's remain constant for each enzyme under the given conditions. This value would be a constant, and it's called Michaelis-Menten constant. So how to determine the Vmax, Km? How to determine this Km? So this curve is actually a little bit tricky. So it's velocity versus substrate concentration graph. So Michaelis Mentes then came up with this equations which tells that the initial velocity of the reaction would be equal to Vmax into K substrate concentration over Km plus substrate concentration for the velocity versus substrate graph for this hyperbolic graph. So now we can use this equation to calculate the values of Vmax and Km. However, while driving this equation, he made some assumption which includes that the substrate concentration of at any given time would be way higher than the enzyme concentration. And this leads to the steady state assumption which says that the enzyme substrate complex would remain constant over the period of time. So during the course of the reaction, because we have huge concentration of substrate compared to the enzyme, so the enzyme substrate concentration almost remains constant during the course of reaction. Thirdly, the initial velocity which we're talking about this V naught figure is being calculated at the very beginning of the reaction, and he assumed that at that point the product concentration would be very low because there is compared to the substrate and the enzyme, and we're talking about the very beginning of the reaction, the product form at that point would be way lower that it can be neglected, it can be ignored, the concentration of the product, because it's close to zero approximately, right? So these are the three main assumptions which he made while driving this equation, Michaelis-Mentes equation, for this hyperbolic velocity versus substrate concentration graph, so in which we can use to calculate the values of Vmax and Km. Although Michaelis-Mentes equation is widely used to explain for the hyperbolic graph between velocity and substrate concentration for any enzyme catalyzed reaction, however, there are some issues with this graph. So one of the problem is that this plateau region is not a real plateau. It goes something like this, and this leads to the error in the calculation of the Vmax value, which in turn leads to the error in the calculation of the Km values. And because the Km is quite an important value to determine the enzyme efficiency, or how an enzyme is how efficient an enzyme is for any reaction, so this error can be an issue, and especially this leads to the issues when we talk about enzyme inhibition or enzyme regulation, because then the small changes or which an inhibitor would be making or an accelerator would be making, it would not be calculated accurately. So this leads to the alright. So then Lineweaver-Burk rearranged this Michaelis-Mentes equation into a straight line equation. And now if we plot a graph over 1 over V naught into 1 over substrate concentration, we get this straight line. And the intersect on the x-axis give us minus 1 over Km value, and intersect on the y-axis gave us 1 over Vmax values, and the slope of this curve or this line would give us Km over Vmax. So this made things a bit easier, because now a effect of an inhibition or an inhibitor can easily be identified, because the line would either shift left or upward or downward direction, and we can even identify the small change in the enzyme activity, right? All right, so now as we have already done the Michaelis-Mentes and Lineweaver-Burk plots, let's talk about the factors which we should consider while looking at an enzymatic reaction. All right, so one of the key factor which affects the enzymatic reaction is the substrate concentration, and this is a graph which was explained both by Michaelis-Mentes and Lineweaver-Burk equations earlier, and this which tells us the significance of the substrate concentration in determining the reaction rate or the reaction rate of an enzymatic catalyzed reaction. And the graph tells us at the initial stages this velocity is increasing with the substrate concentration, and later on, as the substrate concentration reaches to certain threshold values higher than certain, this graph plateaus off. That means the velocity is no more changing with the substrate concentration, and this is because in this region all our enzyme is being occupied. So all of the enzyme has already bound to the substrate, so there is no enzyme left for the substrate we are adding additionally. So the velocity, the reaction has reached to its maximum velocity, and now even if we add more substrate, because the enzyme is not available, there is no free enzyme available to act on the additionally added concentrations. The velocity doesn't change with that time. So this tells us the how an substrate concentration would affect the reaction rate of an enzyme catalyzed reaction. Then another important factor is the temperature. So enzymes are protein in nature or RNA in nature. They are very sensitive to the temperature. So for most of our body enzymes, human body functions at 37 degrees centigrade. So for example, most of our body enzymes have optimum pH or maximum activity somewhere here at 37 degrees centigrade. And below 37 and above 37, the enzymes wouldn't be functioning at its optimal activity. So this optimal temperature can be different for different organisms. For the example, the bacteria who are found in hyperthermal vents would have a different optimum temperature for their enzymes. Similarly, enzymes are sensitive to the pH. So the pepsin, which is a stomach enzyme, works at a very low pH, while the amylase, which is a salivary enzyme, works around 7 degrees centigrade, because our saliva has a temperature around 6.8 or 6.9, so its optimum temperature is in that range. And then alkaline phosphate, it works around 9, pH 9 and 10. Of the optimal pH is there because the enzyme is most active in the bones, and its optimal pH is the pH which bones have, and which is slightly basic. So it works around pH 9 and 10. Right. So these are the key factor which affects any enzymatic reaction, and if you change those, the activity of an enzyme would be affected. Regulation of enzymes. Regulation of enzyme is a very important component of any living system, because all our reactions are being catalyzed by enzymes, and these enzymes need to be regulated for the optimal functioning of any living system. So there are various mechanisms through which the living systems or our bodies are regulating its enzymes. One of the important factor is allostatic regulation. So in the allostatic regulation, most of the time substrate itself acts as an allostatic regulator. These enzymes or proteins have multiple binding sites for their substrate. For example, the hemoglobin has a 4 binding site for the oxygen, and what happens is the binding of the first oxygen molecule is less efficient compared to the second one, and the second one is binding the second one is less efficient compared to the third one, and the third one binds more easily than the first two ones, and the fourth one binds the most efficiently. This is because the binding of the first oxygen brings about the conformational changes in the hemoglobin protein in a way that the binding of the second one is increased, or the affinity for the second one increases, and same goes on. So the fourth one binds most efficiently, and because of this, we have the initial slower rate with the first oxygen molecules are binding a bit less efficiently, and then because the subsequent bindings are more efficient, we get this increase in the velocity of the reaction. And finally, as for any enzymatic reaction, after a certain substrate concentration, this curve plateaus off. So we get this sigmoidal curve, and the regulators, the accelerator for this kind of a reaction would move this curve to the upward direction, and the negative inhibitor would move this curve to the downward direction, right? Another important mechanism for the regulation of enzyme activity in our body is feedback mechanism, and this tells, because of this, for example, this is a metabolic pathway it comprised of multiple enzymatic reactions. The end product gives a feedback and inhibits some of the enzyme which are present earlier in the pathway when there is enough product available. So the body if the end product is enough as the requirement of the body, it will go and inhibit the process at the early steps to stop this pathway. So this mechanism is used in many metabolic pathways in our body to tell when the product is available in enough quantity and we need to inhibit a reaction. And sometimes if a product is not in enough quantity, the reaction would accelerate or go further. For example, if we have enough ATP available in our body, so the glucose breakdown, glycolysis, ATP, TCA cycle, and electron transport chain would be inhibited, would be slowed down because the enough energy is already available. On the other hand, if ADP is available and we have a deficiency of oxygen ATP molecules, these pathways would be accelerated because now we need that energy and the enough ATP is not available. So the ATP, which are the end product of these metabolic pathways, are actually giving a feedback. They are telling the system when these pathways should be activated, should go on and make more and more energy, and when they should be stopped. So in case of deficiency of ATP, the glycolysis, TCA cycle, and electron transport chain would be enhanced because we need ATP. But in the presence of enough ATP molecule, these pathways would be slowed down and we might have the glycogen synthesis pathways going on. So we are storing the glucose instead of utilizing it because we already have the enough energy. So the feedback mechanism is the key regulatory mechanism in the living system. Then come the covalent modifications. And in the covalent modification, phosphorylation and dephosphorylation is the key mechanism where the phosphate group is either attached or removed covalently from any enzyme. The kinases are the protein or the enzymes which add phosphate group covalently to the enzymes. And the phosphatases are the enzyme which remove this phosphate group from other proteins. Okay, so some enzymes are activated by the phosphorylation, some are inactivated by the phosphorylation, some are activated by the dephosphorylation, some are inactivated by the dephosphorylation. An example is glycogen phosphorylase. Its activity is enhanced by the phosphorylation, whereas the glycogen synthase activity is decreased by the phosphorylation. So this covalent modification is a key mechanism to regulate the activity of the enzyme in living system. And finally, another important regulatory system is synthesis and the breakdown rate of the enzymes. So the synthesis and the breakdown rate actually controls the quantity of the enzyme or the amount of an enzyme present in any system or any cell. and it's been tightly regulated. All right, so if a pathway is need to be accelerated, then maybe the enzymes required for that pathway would be synthesized more, and their breakdown would slow down. For example, insulin in the presence of high glucose concentration cause an increase in the synthesis of the enzymes involved in glucose metabolism because they are needed now. So the insulin is present, and it's sensed that there is a huge glucose levels available now, and the body should synthesize more enzymes which are involved in the glucose metabolism, right? So this actually gives another layer of control or regulation for the enzymes in our body. Enzyme inhibition is a very important component of studying the enzymes. A lot of our drugs are enzyme inhibitors, and so it's really important how an inhibitor would affect our enzyme activity. Similarly, our body itself uses a lot of molecules as inhibitor of enzymes to regulate their activity. So enzyme inhibition can be divided into two broad categories, which include irreversible inhibition, where the enzyme is permanently inhibited, and reversible inhibition, in which the enzyme is reversible, so it can be restored, it can restore its activity. Reversible inhibition include competitive, non-competitive, and uncompetitive inhibition, and we'll go in detail in each one of them in coming slides. Irreversible inhibition. In this kind of inhibition, the inhibitor binds to the enzyme covalently and make a complex in such a way that enzyme remains in an inactive form. So usually these inhibitors are either substrate analogs or a transition state analogs, and no enzyme substrate complex would form in case of the irreversible inhibition, because your inhibitor and its active your enzyme and its active site is bound to the inhibitor molecule, and it's no longer available for the substrate binding. This kind of inhibition is sometimes called as suicide inhibition because your inhibitor itself is being consumed in this process, and it's permanently bound to the enzyme. An enzyme remains in its inactive conformation and no longer available for the reaction. Augmentin. Augmentin contains a penicillin group of antibiotics, amoxicillin, and as we know that penicillin has a huge resistance in the population, but we are still using augmentin and it's effective. So what's there? What is in augmentin which makes it effective? So augmentin has two components. Augmentin has an amoxicillin, which is a penicillin group of antibiotics, and then it has an inhibitor molecule, which is culvinanic acid. This inhibitor is actually inhibiting the beta lactamase enzyme. Beta lactamase enzyme is produced by the bacteria to break down the antibiotics, and it's a major cause of antibiotic resistance in the bacterium. So augmentin uses this strategy, that it uses a penicillin based antibiotic, but the inhibitor it contains inhibits the resistance, this enzyme which is the cause of the resistance. So now this beta lactamase, even if it's present in the bacteria, it is completely or irreversibly inhibited by the culvinanic acid, and it would no longer be available to break down the antibiotic, and an antibiotic can go on and function. So that's why augmentin is effective. Another important tannalic acid. This was a diuretic agent and it was available in the market, but later on it was found that it is actually the inhibitor of cytochrome P450, which is an irreversible inhibitor of cytochrome P450. And this cytochrome P450 is a key enzyme involved in the drug metabolism and xenobiotic metabolism. So this drug needs to be retracted from the market because it's actually causing a lot of side effects by inhibiting the cytochrome P450, which is needed for body to function normally. Reversible inhibition. Within the reversible inhibition means the enzyme inhibitor can bind to the enzyme and then it can actually release the enzyme as well. It has three different types. One is called competitive inhibition, in which the enzyme competes with the substrate for the active site binding. So the both enzyme and substrate have the same binding site, which is the active site on the enzyme, and they compete for the binding. The second form of the reversible inhibition is non-competitive inhibition. In the non-competitive inhibition, the inhibitor binds at the regulatory site, but by the binding of the inhibitor, the enzyme is affected in such a way that the conformation of the substrate active site changes and it can no longer bind to the substrate molecule. Right? So there is no computation for the active site, but the conformation of the active site is changed by the binding of the inhibitor on the regulatory site such that it's no longer available for the substrate. Then comes the uncompetitive inhibition. Uncompetitive inhibition, the inhibitor binds on the regulatory site and the conformation of the substrate active site is such that it can bind the substrate, but the reaction, it cannot catalyze the reaction. As long as the inhibitor is bound to the enzyme would not be able to catalyze the reaction. So in this form, although the enzyme substrate complex has been formed, but the reaction is not going forward in the forward direction because the enzyme is no longer in a position or in a conformation to catalyze that reaction because of the presence of the inhibitor. In the case of non-competitive inhibition, we have seen that no enzyme substrate complex has formed. In case of competitive inhibition, there is some enzyme would be bound to the inhibitor molecule and some enzyme would be bound to the substrate molecule because they are competing with each other. So some would be bound to the inhibitor, some would be bound. So there would be some enzyme substrate complex available in the reaction mixture. Right? So let's have a look how this reversible inhibitors affect the reaction rate or reaction kinetics in the next slide. Competitive inhibition, non-competitive inhibition, and uncompetitive inhibition here. And let's have a look how each one of them is going to impact the enzyme kinetics. So in case of competitive inhibition, you can see here the both substrate and inhibitor are competing for the active site. Alright, so as we will increase the substrate concentration, it will eventually compete off the inhibitor and it will attain the same Vmax as the normal uninhibited substrate would do, but it will take a bit longer, so the curve has shifted downward. However, the Km would increase. That means in order to achieve the half Vmax, we need higher amount of substrate. The substrate affinity is bit decreased. This is because the substrate is not just binding to the active site. It has to compete with the inhibitor to bind to the active site. And at a very high substrate concentration, it will compete off the inhibitor finally, but it will take longer time and the Km in this case would be increased. That means our affinity for the substrate for the active site will be reduced, and the same can be represented by the double reciprocal or Lineweaver plot here, that Km is increased but the Vmax remains constant in case of competitive inhibition. In case of non-competitive inhibition, what happens is, because now the inhibitor is bound to the enzyme and the substrate, the active site is such that it's no longer binds to the substrate. So as it binds to the inhibitor, it is not available to the substrate. So no matter how much substrate you add, some of the enzyme would always be bound to the inhibitor because they are not competing for the same site. Alright, so some of the enzyme would be bound to the inhibitor and the active site would not be able to substrate. So your Vmax would be reduced. However, the Km in this case remains the constant. So remember one thing, but this in this case half Vmax has also changed. So half Vmax has also reduced. So we have lower Vmax in case of inhibition, and we'll have lower half Vmax. However, the substrate concentration remains the constant. Km value remains constant, so the substrate concentration to achieve this lower half Vmax is the same as in case of non-inhibition, right? And this is also because this represents the affinity for the of the substrate for the active site has not changed, only because when the inhibitor is bound, this active site is just not available, and we have lower Vmax and we have lower half Vmax, and to achieve the substrate concentration to that lower half Vmax, the Km is the same, right? So substrate concentration to achieve half Vmax remains constant, but our half Vmax is now lower compared to the normal graph, and our Vmax is also lower because some of the enzyme is bound to the inhibitor and never be available to the substrate. And the same can be depicted by the Lineweaver plot. Your Km remains constant, but the Vmax has lowered down. In case of uncompetitive inhibition, what happens is the inhibitor binds to the enzyme substrate complex, right? So, but what happens is the reaction doesn't proceed. Okay, so in the same case in as in the non-competitive inhibition, because some of the enzyme would always be bound to the inhibitor, and no matter how much substrate concentration you add, because they are not competing for the same site, some of the enzyme would not be available, so the Vmax is reduced and the half Vmax is also reduced. But in this case the Km is increased. This is because that means that shows that, oh, the affinity of the enzyme for the substrate increased. And to some extent it is true, because when the inhibitor binds to the enzyme substrate complex, it actually reduces the probability of substrate dissociation. So substrate dissociation probability reduces, which actually can be reflected here as a decreased Km value. But remember, as in the case of non-competitive inhibition, your Vmax has reduced and your half Vmax has also reduced. So this Km calculation is for that lower half Vmax, and it is lower also because the dissociation of the enzyme substrate complex uh, is now a little bit more difficult in the presence of the inhibitor, because inhibitor kind of stabilize this conformation. It's actually not going backward or not letting the enzyme to go forward or complete the reaction, right? So that's why we have lower Vmax and lower Km in case of uncompetitive inhibition, and the same can be seen here in case of non uncompetitive inhibition and the Lineweaver work plot or rubber reciprocal plot. Okay. So now let's have some examples of each one of them. So sulfa drugs are the ones which are involved in the which are the competitive inhibitors of PABA, and PABA is a precursor which bacteria use to produce their folic acid. So sulfa drugs are used as competitive inhibitors. So humans cannot produce the folic acid, so we need that in our diet, but bacteria uses their metabolic pathway where the PABA is a precursor to produce a folic acid. So we can use a competitive inhibitor of PABA as a drug as an antibiotic drug to kill the bacteria. Statins are also which are used to lower the blood cholesterol levels, or also the competitive inhibitors for the enzyme CoA reductase. Non-competitive inhibitor as represented earlier and shown earlier, that they actually Km remains constant, the Vmax increases in this case, and our cyanide which actually binds the cytochrome oxidase at allostatic binding site and inhibits the electrons from being passed on the electron transport chain, is a non-competitive inhibitor of cytochrome oxidase. Heavy metal poisoning, in which the mercury and lead goes and bind to the SH groups of the proteins and enzymes, and allostatically and non-competitively inhibit their activity. Uncompetitive inhibition, as mentioned earlier, in this case both Vmax and Km is decreased, and the inhibitor bind to the enzyme substrate complex. Our oxaloacetate is an example of this kind of inhibition, where oxaloacetate inhibits enzyme lactate dehydrogenase. So oxaloacetate, if the enough quantity of oxaloacetate has been produced in the TCA cycle, it actually goes and inhibits earlier steps of glycolysis through uncompetitive inhibition. Mixed inhibition. So some enzymes have multiple substrate. For example, they have two substrate and two binding sites. So they are two active sites. And what happens in case of mixed inhibition, that the substrate 1 binds to its active site and there is no competition for the active site of the substrate 1, but the inhibitor competes with substrate 2 for its binding site. So there is a competitive inhibition for the site 2, but there is no inhibition or competition for site 1. So in this kind of cases, we observe the phenomena of mixed inhibition, and in case of mixed inhibition, you can see the competitive inhibition graph moves in such a way that we have lower Km values and lower Vmax values, and higher Km values.